Good afternoon, everybody, and welcome to this month's NHIP seminar. So Newcastle Health Innovation Partners is our academic health science centre, as probably you know by now if you've been to these seminars previously, but it's a partnership between Newcastle University, Newcastle Hospitals, HYNINC, Health Innovation North East and North Cumbria, our local mental health trust, Cumbria, Northumberland, Tyne and Wear NHS Trust, and Newcastle City Council. And I chair the Research and Innovation Strategy Group, and the role of that group is to really promote cross-disciplinary collaborative research between the various NHIP partners. And advanced therapies is one of the areas that we chose right at the beginning of NHIP as an area of multidisciplinary science, which would benefit from the partnership. So being more specific, this is our, as I say, this is our monthly, or in fact, 2 monthly advanced therapies seminar. And again, the idea of the seminar series is to socialize the really great advanced therapies science that goes on around Newcastle, around the region, which is often invisible to people who are not sort of encountering it daily. So that's all just by way of background. But the most important thing for me to do is introduce today's speaker, and it really is a true pleasure to introduce Kenny Dalgarno. I've known Kenny pretty much since I arrived in Newcastle 22 years ago, particularly around work in nanotechnologies when Andrew McCaskey was developing nanotechnologies in Newcastle, and I think that's when I first met Kenny. Kenny is the Sir James Woodeson Professor of Manufacturing Engineering at Newcastle University, and he's the lead for the Newcastle University Centre for Biomedical Engineering. He was Deputy Director with Andrew McCaskey, actually, of the Versus Arthritis Tissue Engineering Centre. That was a very successful centre of excellence. And he's also the Newcastle University lead investigator for the EPSRC Centre for Doctoral Training in Additive Manufacture and 3D Printing. Ken has been researching the use of 3D printing and additive manufacture for biomedical applications for over 20 years, with an increasing focus recently on bioprinting and biofabrication for regenerative medicine and disease modelling. And that work's been supported by the EPSRC, European Commission, Arthritis UK, Versus Arthritis UK, I should say, the Carbon Trust and industry. Ken is also the Chief Scientific Officer of Jetbio, a bioprinting spin-out company which he's developed from Newcastle University. So as I say, I'm really looking forward to today's seminar, and I hope you are as well. I think we're in for a real treat. So Kenny, over to you. Thank you very much for offering to provide this month's seminar.
That's great. Thanks, John. I'll get what I'll do. It's lovely to be here this afternoon. What I'm going to do is just share my screen and get the slides up and check that everybody can see what they're able to see.
Yeah, we can see that, Kenny.
So we're to go onto the slideshow. That should all come across. That's good.
That's good.
So yes, what I have for you this afternoon is a talk on bioprinting and biofabrication for in vitro models. Clearly, lots of advanced therapeutics need in vitro models for evaluation. I guess it's something maybe a little bit different, but hopefully interesting nonetheless. So if I just sketch out what I'll talk through, I'll do a quick introduction, if you like, to models and how we think about them and biofabrication. Do a little lesson from leukemia, delivered by Turing nanotubes, which will hopefully make a little bit more sense once we've done that. Talk then about the reactive jet impingement bioprinting process that we've developed here, and then just show you some examples of ongoing work that we have using that process and some other interesting bits of technology. Okay, so I'll kick off first of all with models, and I guess with in vitro models, physiological micromodels, lots of different names from them. In engineering, we nearly always revert to the view of George Box, who's a famous American statistician, He said that all models are wrong, but some are useful. There's kind of seeking perfection in a model isn't really the thing to do. You want to make sure that it's useful. And what we know collectively is that lots of in vitro and in vivo models of many diseases lack predictive capability for drug development. There's lots of good work can be done in terms of safety, but if we had more predictive capability for drug development, then 9 out of 10 drugs wouldn't fail when they got to clinical trial. I guess would be the main headline. And so what we're doing is seeking more useful in vitro models as a general rule. And we tend to interpret that as looking for models which are closer to the physiological environment. Those of you who are in the field, you understand there's lots of different formats for the different models that we use. We can have, we could have 2D monolayers in well plates. You can arrange your cells into 3D clusters or spheroids. You can then have matrix, so extracellular matrix components or a sort of a synthetic equivalent. You can have cells on top of matrix or within matrix or simply in fluid. This is just a quick picture which doesn't encompass even half of the different options you have for how you go about them. And then there's also options in terms of, if you like, the overall packaging of a model. In drug development, because people want to head quite quickly towards relatively large scale. So 96 well plates, 384 well plates, and do things on a sort of industrial scale. And certainly that's where a lot of the interest is from pharma companies. But the other area that's been growing a bit that's almost the opposite in terms of scale up is the kind of lab on a chip or organ on a chip or body on a chip type of activities where you have a set of complex interactions and you try and make all of that happen in a sort of miniaturized form within a sort of a single package, so that you bring all of this together in a sort of chip-based assay. So there's lots of different ways and means that this can look. But engineers, we always like to have a kind of process view of the world. And so although it's lots of different formats and they all look very different, the actual way you go about it tends to be very, very similar for all of these. You have some cells, which is what you're normally basing your in vitro model on. You need to use these to establish a culture of some description. One of the examples we saw on the previous page. You then need to let that culture mature, so that you get the cells behaving as they would physiologically. You have a micro model then at this point, once you've got your tissue mature. And as I say, it's a model. It won't replicate everything to do with your organ of interest, but it should replicate some of the key components that are useful to you. And at that point, you can introduce your compound or therapy through the downstream analysis, and on some occasions that will result in a treatment being identified. And so that kind of whichever of these formats you're in, this process tends to also apply. And all along that kind of scope, as you go along here, there's lots of what we refer to as liquid handling. So you can have active and passive pumping, whether it's actually with pumps or with pipettes or whatever it happens to be, nozzle-based dispensing. So you need to, throughout this process, essentially, you need to move a lot of fluids around, in and out, in terms of media and therapy itself and all those kind of things, and preparing for analysis. And so with that kind of overview, the kind of question was, well, where actually can bioprinting and biofabrication help in terms of this overall process? And kind of mostly it's at the beginning and in the liquid handling. If you need to put together at scale cultures which are quite complex, then that's really where bioprinting comes into its own. Biofabrication techniques can help with if you want to mature your tissue quicker. So get to a functional tissue that you can use in some way as quickly as you can. Then between the bioprinting and biofabrication, you can kind of accomplish these two activities. And then with liquid handling, yes, that automating that is a sort of bioprinting type of activity. And it tends to be that the imperatives for scale up are that we want these cultures to be as small as they can be and as simple as they can be, but we want the whole process to be integrated and automated. So that's where we go. So that's where we're kind of starting and where bioprinting, that's, if you like, some words on models. If I now talk about biofabrication and bioprinting to a greater degree. So biofabrication, it's kind of, if you Google biofabrication, you get a picture, get this picture or one very like it, where you have an organ that's been printed. And yeah, it's interesting. It's representative of the thing that we absolutely cannot do at the moment is print an organ. But it does give you it's a nice image in terms of getting the central idea across that really what we're about is processing biological and synthetic materials to try and create a structure that has a function, and the designed biological function is the key part of this. And the heart picture does also take us to often when people talk about biofabrication, they are talking about tissue engineered implants, whereas this afternoon the main focus is more on using these technologies for kind of disease modeling, toxicology, drug screening, diagnostics, theranostics, in vitro applications. So it's about sort of putting together complex cultures, I guess you might say. And then there's if you're new to biofabrication, there's a lovely review paper by Murphy and Atala in Nature Biotechnology from a while ago, where they broke it down into a kind of 5 or 6 step process. They said essentially, to start off with, you have to decide what kind of shape you're going to make. If you're having a kind of material as part of your culture, you need to decide what that's going to be, whether you have a synthetic or a natural polymer, or you use extracellular matrix. Again, I'm in a sort of vague way, which is as in I'm talking about cells as a sort of in general type of thing. But for your application, you have to decide what's going to be the best way to acquire, mature, and develop your cells. Then there's a step they've got bioprinting, which is essentially where you put this all together. You take the shape that you want with the material that you want, cells that you want, and you create your kind of complex culture in some way. The important thing, as I'll say to students, the important thing about bioprinting is that it's not actually the end of anything. That just gets you a starting culture. It then has to be matured and applied. And so it may be that it goes into a bioreactor to be matured, or more likely directed to implantation if it's for tissue engineering, or as we're looking at this afternoon, something for in vitro testing. And so it's that's the kind of view of how a kind of biofabrication process works. And as part of talking about biofabrication, I'm going to focus really on 3D cell culture in hydrogels. And a few kind of reasons for that, and we'll kind of get through it in a minute. But it's now relatively well recognized that 2D culture isn't always the most representative of how cells behave in tissues. You don't have the same kind of cell-to-cell and don't have the same kind of cell-to-cell and cell-to-extracellular matrix connections in 2D culture as you do in 3D culture. And as we'll see in a minute, sometimes that can lead to key mechanisms being mixed. So 3D cell culture, although it's more difficult in terms of downstream analysis, applications much more representative of what actually happened. And then hydrogels are interesting because they provide a sort of an aqueous, biocompatible 3D environment for cells. You can arrange for them to be structurally similar to soft tissues. You can then introduce into them proteins, pharma agents, other biochemicals in the same way that you can just a fluid. And so that they have a lot of advantages in terms of essentially giving quite a biomimetic environment for cells. And but the one thing I'll note, because it's something I'll come back to later on, so this is a bit of foreshadowing, if you like. Many of you might have made jelly, or you might have made gels in some way, but with the materials we work with, the gelling reactions generally involve taking two solutions, a polymer solution, a cross-linking solution, something like that, where you have, and they're both close in viscosity to water, so they're quite liquid solutions, so much more water than oil or gel. And then you use a chemical reaction of temperature or pH to cross-link, and that produces a little high viscosity gel, as we go down here. And so you end up with a little gel droplet, or a jelly tot, if you're of an age to remember what a jelly tot was, which has, and if you've encapsulated cells within it, then you can have cells within here as well. And so, yeah, we have ways of creating gels, but the main thing to note in processing of them, which I'll come back to with my engineering hat on in a bit, is that you start off with relatively easy flowing fluids, and you end up with something that's really quite stiff and can be difficult to make it. Okay. So then I promised you a sort of a story from leukemia. And really it's to do with how leukemia cells evade drugs, which is a distinct problem in drug development. It's one that's been done by Sean Hockney, who's a PhD actually at Northumbria, but is also a visiting researcher here, and I co-supervise him. And Dipali Pal, who was based in cancer here, then moved to Northumbria, and has recently just moved to Bristol. And so what we have up here is essentially it's a leukemia co-culture. We have a mixture of cells in red, which are acute lymphoblastic lymphoma cells, ILs, and then we also have stem cells, but bone marrow derived stem cells, so stem cells that would reside in the bone marrow niche where leukemia arises. And so the stem cells are nuclear stain, which is green. The leukemic cells have nuclear stain, which is red, and we can separate out the images or look at them together. I'm sure most of you will understand that. And what they did, so this is kind of normal culture, standard development of this co-culture. And then dexamethasone is a steroid, sometimes used in the treatment of cancer and is used in the treatment of leukemia. If we introduce dexamethasone into this co-culture, I see these lines appearing between the nucleus of the cells, and it's carrying this nuclear stain that's been applied. And these are the tunneling nanotubes that I was on about, and I find this fascinating that there's that one of the ways that cells can transmit information between one another is to have a direct tube that's created between the cell nuclei. But that's what's happening here, and we see so. And the story, if you like, we think, is that the dexamethasone is introduced into this culture. The cells start to feel stressed about the steroid, and then as a result of that, they start communicating from cell to cell. And you can see the stem cells communicate with one another, leukemia cells communicate with one another. If we look at the two together, though, there's a slightly more interesting picture. You start to pick out some bits in yellow here, where clearly there's it's not stem cell to stem cell, it's not leukemia cell to leukemia cell, it's crosstalk between the two. And it's a lovely little visual way of picking this up. And so if I go back to my question at the top, how do leukemia cells evade drugs? Well, what we think this is saying is that okay, the leukemia cells start to feel stressed when you introduce the drug or the medication, and one of the things they kind of transmit out through the tunneling nanotubes to other cells in the nearby location, and some of those will not be leukemia cells; they'll be essentially healthy cells, because the drug is targeting the cells that look like cancer cells. It's essentially a way of the disease to hide itself in amongst healthy cells, or in amongst cells that currently look healthy, and that gives it essentially a way of hiding the disease from the drug for a while, and sort of evading the effect of the drug. And it's just a beautifully kind of visual way of seeing this. I think it's a lovely piece of work. The biology understanding of it is much better explained on this paper that Sean's put on bioRxiv. It should come out soon too. But yeah, once you understand this mechanism, you can then do things, look at other drugs. So this is ADH1, ABT199. These two newer drugs, but for both of them, we still see these tunneling nanotubes appearing. We still see bits of yellow, so there's still sort of evasion of the disease happening. But then you can look at drug combinations. So DEX with ABT199, not that much more effective than either of them alone, because you can still see all of these lines going through. DEX with ADH1, however, gives us very little. And so the kind of story here is that for this particular strain of leukemia, that maybe this DEX ADH11 is the right drug combination to try, and not only attack the cell, the leukemia cells, but to stop them evading the effect of the drug in the first place. And so, yeah, so there was a lesson in here, but and the main kind of thing here is it kind of reinforces the need for these 3D hydrogel based models, because these tunneling nanotubes, first of all, they have to have matrix or something matrix like in order to tunnel through, or else they just won't appear. And secondly, the other thing is really to do with cell density. If the nuclei aren't close, then the nanotube effect kind of doesn't happen. And so if without kind of a really good start 3D model to start with, you miss this mechanism entirely. And so it's really the lesson from leukemia is that in 3D cell culture, the cell density and having some kind of representative matrix are really important in ensuring that you get the right cell-cell and cell-ECM interaction within the models, and picking up on those specific mechanisms. And then the other point is, yes, if you have a proliferative cell line to make your models from, then you can start off low density and end up high cell density, but that takes time, and our experience is that people don't like spending lots and lots of time culturing. So if you need to wait for weeks, then already the drug companies are starting to kind of look at their watches and consider what they might be doing better over time. And something that's high throughput won't need lots and lots of time to develop. So then, yes, so having kind of made the case for this type of cell culture, we look at how we actually bioprint gels and get into the different bioprinting processes. So if you've done anything with bioprinting before, it's common that you've used a micro extrusion bioprinter, and this is one where you have something that either is a syringe or looks like a syringe, and you produce a gel, cell-filled gel, and you apply pressure on the back in order to kind of squeeze the track through your needle, and then you can create a pattern from it. And that works very well until you get to a certain viscosity. And I said earlier on I was going to come back to viscosity. Clearly, the thicker your fluid gets here, the more pressure you have to apply on the back of it. And as you build up the pressure, you build up shear stresses on the side, you build up stresses at the peak, and so you get to the stage where actually you can be killing quite a lot of your cells, which is of no use to anybody. And so people at that point tend to back off in terms of viscosity, but you then end up with something that's quite close to water. And so the more cells you want to put in, to some extent, that gives you limit in order to get the kind of correct cell-cell ECM interaction. The more difficult it is to have a gel which is at the stiffer end of the gels. And there's a number of ways of going about doing this. You can do some of the chemistry on the substrate, if you like. So have something relatively low viscosity and then react it. But it is still a kind of a problem. The lower your viscosity is, the higher your cell density can be. But it does tend to be the lower your viscosity, you don't get the same level of accuracy, or you need to do some kind of post-print curing, as it's called. It's a kind of like a technical point, but it's nonetheless. And then, so one of the ways that people started to think about avoiding this problem was by what we call late gelling. So rather than making your gel and then extruding it, the idea was that you'd have your gel precursors, which are, so in the blue and the red, would might be your polymer solution and a cross-linking solution. And you'd bring them together at the very end. And so the chemical reaction happens here, and the gel is produced down at the bottom. And that can work quite well. There's a Canadian company called Aspect Biosystems who have a nice example of this. And so that comes through. But there's a kind of, it's more or less a sort of continuous process, though. It's difficult to turn on and off because by the time you've got your thing here actually being gel, if you then stop what's going around, you just kind of plug the gap, and it can be. So it needs careful thought. So the process we developed at Newcastle is one called reactive jet impingement, where we can take that late gelling approach one step further, if you like, and move it outside of the cartridge altogether. It's a slightly kind of complicated process to describe. I'll kind of do my best. What we have is what we call two microvalves, and we generate droplet streams from these two microvalves. One of them is cells in a cross-linker solution, and one of them is, if you like, the polymer solution. So we have these droplet streams from these two fluids, and where we arrange for them to meet in midair. Where they meet, they react and form these little droplets. And if we've suspended cells in one or other of the solutions, then we get the cell-filled hydrogel droplets generated, and they then fall into the substrate. When it's working, it kind of looks like this. So this is microvalve here and microvalve here, and this kind of collision is happening here, but you can't quite see it, and then you see the droplets landing on the substrate. And so it's just a very, very nice way of making small gels that can be cell-filled gels, and we'll see the advantages again in a minute. So that's the kind of process with the two fluids and the reaction. You couldn't see the actual collision of the droplets in the previous slide, but you can with high-speed camera. We've also done some modeling, mathematical modeling of the process to see how these fluids combine. And we can generate kind of droplet arrays, as you saw, or 2D or 3D or any kind of 3D bioprinted type of shapes as we go along. And it's something, yeah, as I said, it was developed at Newcastle. We patented it, and with myself and Matt Benning as the joint inventors on the patent. And we do now, as we mentioned in the introduction, we have a spin-out company called Jetbio, which is just looking for seed round funding in order to start commercializing the technology. I'm not going to say a tremendous amount about materials, but what I will just say, because this could be another hour-long talk in itself, But the gel that we use most often currently is one we call CAF. It's a gel produced from collagen, alginate, and fibrin. And those three materials, the alginate and the fibrin, give us a quick cross-linking. We need the reaction between the two fluids to happen very quickly. And then the collagen and the fibrin give us biofunctionality. So they all work very nicely together. And we found this to be a really good kind of general purpose gel for cell culture. When we need to, we can tune it in particular directions. So at the moment, we're doing some work with cardiac. We're looking at combining types 3 and 4 collagen with collagen 1 in order to be more biomimetic. And so it gives us a really good foundation that we can tune towards most applications. So I won't say, so it's just, it'll be CAF gel, and I won't labor that point any further. And then when we started off, it was, again, as John said in the introduction, work that was funded by Versus Arthritis. And so we were interested in looking at bone microtissues. We wanted to create those. And so the way we've gone about this, we have our CAF gel. So we have the polymer components on this side, coming in from this side. Over here, we then have the thrombin, cross-link the fibrinogen, the calcium chloride, cross-link the alginate. But in this side, we've also have human mesenchymal stromal cells, Y201s. And when we printed them, what we found is that we could get very, very high cell densities. So we could get these kind of 45 million cells per milliliter of gel. And this is just a bright field image where you can see these cells and in the kind of hyperconfluent gel. And so that was interesting because that goes well beyond what other bioprinting processes can do. So we wanted to understand actually how meaningful is it at this point. We now sort of recognize it a bit more, but at the time we wanted to understand really is this a valuable thing. So we had an experiment where we had what we called a low cell density gel and a high cell density gel side by side. This is 2 million cells per milliliter of gel. This is 20 million cells per milliliter of gel. And you can see that this one's quite sparse and this one's quite busy. You can also see, sort of like that, is that the higher cell density gel gets itself a bit more organized later on. These cells are growing up and work fine, but there's a bit more organization in the culture down here. With bone microtissues, what you look for is mineralization and matrix formation. So these images are looking at here. You see these nodules that you see are calcium deposits, and so actually I should say so these MSCs have been cultured in osteogenic media. They then turn into osteoblasts and deposit the calcium. And so this is actually quite nice calcium there, but with the high cell density there, we've got a lot of calcium deposit out there. And again, we're still seeing more kind of organization in the little microtissue. So in terms of kind of benchmarking, I mean, what this is kind of suggesting to us is that the more cells you have in, the more mature your culture is, which is good. But we looked at that also then biochemically. So we have a PCR array where we're looking at a range of osteogenic markers. And on this axis, full change, we're comparing the high cell density gel to the low cell density gel. In terms of these markers, osteopontin is SPP1, and then we have bone morphogenic proteins 2 and 6. And these three are all sort of indicative of relatively late stage osteogenesis. So these are telling us, confirming what we saw really, that the high cell density culture is maturing to a greater degree than the low cell density. But the other thing that it tells us is that the effects of increasing cell density aren't linear. So yes, we do have 10 times as many cells, but in terms of the expression of proteins, we've got kind of 50 times, coming out. And so we seem to get more from each cell in terms of the bioactivity. as you put more and more cells together. And so cells stimulate one another, if you like, in the tissues. And so that was kind of interesting for us, and that kind of took me back to this picture, that was from this Murphy and al. The biofabrication. I guess the point I really want to make here is that the only thing I'm not so sure about this picture is that it does set these out as a kind of sequential set of steps which are independent from one another to some degree, whereas actually what material you choose affects how things happen in your application. What cells you choose affects how things happen in your application, and how you actually put them together will affect how things happen in your application, because this is where your cell density and the arrangement of cells relative to one another is all decided. And so actually, this should be more of a loop, because you really start at your application and go back and decide upon, okay, if this is the application, what shape do we need? What material do we need? What cell do we need? What printing approach do we need? And so it just back to this picture to make the point that these stages are not all independent from one another in any way, and that they should all be driven by the application. But okay, so we have this bioprinting process that we've developed for cell-filled gels, rapid deposition rate, high cell densities. We can print 2D arrays, 3D constructs. We can print high cell densities to get our microtissues, and we can print on nearly any substrate as well. And we went on to look a bit more at that, and I'll just quickly go through some examples really of what we've done. So this is a titanium alloy, a medical grade titanium alloy. It's a very rough surface, quite an unpleasant surface to get cells onto to some extent. But we can print our gel successfully, and the cells culture well, and they come out of the gel, attached to the surface, and start to deposit mineral. And this is alizarin red staining, demonstrating that the cells are coming out and generating an interface. Another kind of hard surface, this one's a porous bioceramic. So this is a bone substitute material called apatite molastinite. And here we were looking at using the printing process as a sort of in-theatre cell seeding approach, because within the gel, the cells are immediately attached, and so you can print an implant potentially. But what we wanted to look at was were there any issues with spreading or mineralization as a result of printing the gel on rather than just manual seeding. And what we found is that there aren't, there's no real difference in terms of the seeding approaches, but this one gives you an immediate fixation. We can also make layered cultures. This is kind of, again, versus arthritis. One of our funders, and we had an interest in the cartilage bone interface, and so an osteochondral interface. So what we wanted to make was sort of a culture where we had a kind of cartilage phase and a bone phase next to one another with a tight interface. And when we printed this, so this is at the top half of this is our gel with chondrocytes in. The bottom half of this is our gel with osteoblasts in. I need to show you where the interface is with that arrow, because it's not actually visible, and that's because if we look at it with confocal and labeling the two different cell types, this interface here that we've managed to generate is kind of 2 to 3 cells thick, and so it's a very, very tight bonding across the interface. And so we're moving on now to look at actually can we use that to look at some of the mechanisms that happen at the bone cartilage interface during development of osteoarthritis. We can also print onto tissue. So this is what many of you will know Fibrofind, which is a spin out from the fibrosis group, where they have a cancer invasion model, which is based on on these precision cut liver slices. And the model is essentially one where they have this tissue, which they very carefully prepare, but they then seed some hepatocarcinoma cells onto it and track the invasion of the cells into the tissue. And what we've been discussing and trying out with them is can we actually print these hepatocarcinoma cells onto the little tissue slices? And just to, as a kind of proof of concept, we did this work, and this is a little globule that one you can see there is actually printing onto these precision cut liver slices. And the thing I'll note is they're like 0.25 millimeters thick, they're a quarter of a millimeter, so they're incredibly delicate structures. But the gel doesn't seem to damage them at all, and the deposition doesn't seem to damage them at all. So it's a nice way of doing things. And then we did some very, very preliminary work just to show that using 2-photon, you can track the invasion of the hepatocarcinoma cells into the tissue. And we're now doing some more work with Bioprinting to see if we can develop this up into sort of a way of scaling, of getting more out of each of these very valuable bits of tissue that they have. Then have fibroblasts. This is printing onto fibers. Again, a very, very delicate structure. And just to make the point that we can make these interesting structures. So this is calcium alginate fiber. And these little sponge-like structures here are produced from these fibers. And then when we've printed onto them, these darker areas, we've got focal, are where the fibers are sitting. So what we find is that the gel that we print onto it engulfs the fibers. And so we end up with this is looking at the edge of a structure, we end up with this cell gel fiber composite material almost. And we found this interesting for two reasons. The first one's a very, very practical, simple reason. The fiber clearly is just like a bit of cloth, but it's a really high cell density culture that you can pick up and manipulate and move around and do things with. And so in a practical sense, it's really useful. Also, it gives you a kind of a multi-scale attachment for cells, and that there's the gel itself gives you attachments, and then the fibers into the bargain, and that seems to drive metabolic activity in a nice way. So this is from that previous experiment, and if I just unpick it slightly for you, so where we have green here, we're printing onto cover glass, and where we have grey, we're printing onto the fibers. And then over here, we're at a low cell density, and over here, we're at high cell density. I guess the two main takeaways, the grey is always bigger than the green, wherever we happen to be, sometimes significant, sometimes not. So having the fiber boosts metabolic activity, but also the high cell density is always bigger than the low cell density. And so cell density also drives metabolic activity as well. And so if you want to kind of get the maturation and all your biological processes developing, then having a high cell density together with the fibers is a good way to go. Okay. And so one last example, I'm getting to time, is a cardiac in vitro model that we've been doing as part of a European project. And if I go back to my previous slide where we're at the beginning, where we had a heart, the idea here is to print lots of mini hearts essentially for testing the effectiveness of drugs through a new therapeutic approach. But what I wanted to just show was somewhat did very recently on understanding the functionality of these 3D cell cultures. And we did it using an Accurate 3D chip, and this is something that they sorted out. It's made by a company called 3Brain, and so it's a little kind of well that at the bottom has a load of electrodes, and so you can culture things on the well, and each of these electrodes has the ability to actually deliver or record voltage into the system. And it's the company's called 3Brain. They originally developed it around taking brain slices and looking at neuronal activity. But you can actually just culture cells on these things. I'm sorry, I should have said. So these electrodes all sit at the bottom of this little and so what we've done is printed cardiomyocytes directly onto this structure, cultured them for a period up to a week, and then done two things: done a calcium flux assay to look at cell contraction, and looked at recordings from these electrodes for each sample over a 5-minute period. And just to take it, so that's the chip, and that's our printed gel on top of it. We compared kind of 2D to 3D in terms of it. So, and here, this is the calcium flux assay. So really what this is showing you, where it's fluorescing, is showing you where the cardiomyocytes are contracting, like a beating heart. There's less cells in 2D than in 3D, so the level of fluorescence doesn't tell us much. But what happens in 3D is that these cells start to beat spontaneously, and it's because we've got them close together enough that they beat spontaneously in a coordinated way. And this is really interesting. We don't think anybody else has managed to get this to happen in a 3D gel culture. And so we thought that was really interesting. And then from the electrodes, you get these lovely heat map type of things. So each of these yellow pixels tells you where there's been a voltage recorded as a result of the activity. And what we see is that 2D, yeah, we get things up to day 3, we see activity and then it dies off. And this is actually it's a known problem with these cells, is that between day 3 and 5, they lose contractility in 2D. But it doesn't seem to be the case in 3D. They warm up slower, but once that they're still there and beating after a week, and at each of these little pixels, you get a little ECG type of signal, which is interesting. So first of all, we have a 3D culture where cells will beat spontaneously, cardiomyocytes will, but also we seem to have a system that gives us much more flexibility in terms of the duration of an experiment if we were looking at the incorporation of drugs. And so this is we've enjoyed playing with these chips, and the information you get out of them seems to be really, really interesting. So just to start then, we have increasing focus on human in vitro models. We understand that 3D models can be more representative of kind of cell-cell and cell-ECM interactions for a particular tissue of disease. And then we have our printing process gives us a flexible way of creating gel cultures with the potential for a wide range of kind of cell densities and substrates in order to establish the model that people are interested in. And so I'll acknowledge, those of you who know me will know that I needed a lot of help. And so these are the people who have provided it. I've mentioned some of them as we go along the way, but yes, absolutely thank you to them, and also thank the lovely funders, Versus Arthritis, NCCRs, MMC, and Horizon Europe, who've given us the money to make the work possible. Okay, and so I'll finish there.
Thank you, Kenny. That was brilliant. As anticipated, really, really interesting, actually. Thank you so much. There's a couple of questions already in the chat. If you've got questions, please put them in the chat and we'll try and get through them. The first one's actually from Mark Jarvis. You can probably see the questions yourself, but Mark's question is, with bioprinting, is there a quality control process? For example, is the viability of the cells and integrity of the cells tested in process or final products? That's kind of a regulatory question, isn't it? How will the regulators view these products in practice?
Well, it depends how you were using it. If you're using it for in vitro models, really, I think it then leads to how reliable is it as a process and how similar are you going to get to. But it is one of the reasons for going towards an automated process, that if you've got our process optimized, then we should be able to print the same things yesterday, tomorrow, and today, and get similar answers every time, so that we've got a good kind of base starting model. And so with bioprinting, absolutely, there's a quality control process, but I think only in the same way that there always is. You know what I mean? It doesn't bring a new need for quality control, but it should actually help with quality control.
Oh, thanks, Kenny. Are there any? Is there any guidance? Yes, from the regulators, from MHRA or anything like that about these sorts of products, or are we not quite there yet?
So the FDA has done perhaps more than any of the other regulators, and they've explicitly started to give guidance, and they're seeing it as essentially so the bioprinter, if you like, is a piece of laboratory equipment, and the quality control is on what comes out of it and what you subsequently do to that, if you like. So it's no different from a cell culture robot or in any of those kind of things in terms of how it's viewed. And so it's yeah, as I say, it's a piece of equipment rather than something that's a product in itself.
Great.
I guess, John, sorry, just to sort of expand on that, what I was getting at was just the fact that obviously you're putting a particular cell type in a gel, in a culture, and you're bioprinting that as a product that would be then used against drug testing. Are you doing any tests that demonstrate that the functionality of the cell and the parameters of the cell that go in the gel remain consistent on the same cell type? Obviously, that links with the MHRA that if we were doing a GMP product, in order to take that to market, you would be testing the cellular composition or the viability of that every time. And I just wondered how you, I mean, at bare minimum, how do you test a cell inside a gel in a bioprinted?
Yeah, no, that is a good question. And so there's a number of things in terms of the process that we would tend to in developing a model. You would want to benchmark it against existing models and indeed benchmark it using existing drugs. So you'd be looking for the model to give you the behavior that you expect to see. In terms of getting cells out of a 3D gel, yes, that can be interesting. There's a number of ways on the market for you to do assays on 3D gel structures. Sometimes they work, sometimes they don't. And in the end, sometimes we have to just treat our gel as a piece of tissue and take it to IHC and do things that way in order to get the answers that we want. But yeah, I mean, that's it. So you can do it. Sometimes you have to turn it into a 2D by slicing it in order for it to retrofit it back to our 2D microscopes. But there are some things you can do in 3D. There are some things you can do through the supernatant and the ELISA-like processes or whatever it happens to be. But yeah, do you have to? You absolutely do have to know what's going on, for sure.
Thanks, Kenny. And there's a question from Elisa Villalobos. To do the interface cartilage bone, do you set a defined stiffness in which your cells will be printed, and are the materials different for each tissue?
So in what we've done so far, the materials were the same for each tissue. And that was because we were, if you like, what happening is that the material doesn't actually hang around too long. We'd like the osteoblasts to start generating mineral. We'd like the chondrocytes to start generating gags. And so we'd actually like the material to be overtaken by the ECM quite quickly in the process in order to generate that interface. Yeah, we're not entirely sure if that's happening. And then, so we might come back from that and think about actually printing. We did print onto a sort of a bioceramic substrate. And what we might do is print the bone element onto a hard substrate and then add the cartilage element on top of it in order to more closely replicate that kind of hard soft interface. I hope that got your question, Eliza. But yes, that being something of interest is definitely the case.
Great, thank you. Not got much time left. A very speculative question for me then to finish things off, Kenny. I mean, there's a lot of stuff going on about AI in medicine at the moment, and what's the future of AI? What's the future of bioprinting, do you think? If you look forward 10 years, obviously you've got your own perspective on that, but in reality, where do you think we'll be? What impact will it make?
So I think, again, we've seen from FDA, there was a couple of years ago that the FDA first said, actually, we don't need an animal to approve a drug. And I think that kind of change in mindset is bringing the kind of human in vitro modeling industry, if you like, a bit of a boost. And so I think it is a large scale screening of lots of molecules. And turning that over and being able, and then longer term, if we can get it all to work fast enough, it's about doing that in a personalized way and being able to tune it to an individual. And that would be good. And then on the other side, it's not really been the focus. I mean, I think we, to some extent, for regenerative medicine, there's lots of potential, but the problems in regenerative medicine aren't really problems that bioprinting are going to solve, if you know what I mean. Immunological problems and those kind of things aren't really the case. So if the right cell sources come along, there's potential there as well. But right now, I think it's definitely high throughput screening with if you like, more representative in vitro models.
Yeah. Brilliant. Well, look, we're spot on time. So again, thanks ever so much, Kenny, for a really fascinating talk and another great example of advanced therapies work in Newcastle that's sometimes too hidden from the people that need to see it. So maybe we'll get you back again in a year or so to tell us where you've got to at that stage. So thanks ever so much for joining us, and thanks to the audience. Thanks for your questions, and see you all again soon. Thank you very much.
Thank you, John. Thanks all.
Bye-bye.